In this video, we're going to be building on the previous one where we drew each structure of the natural amino acids at a fully protonated state. But now we're going to look at how pH, and I've selected 7 here, is going to influence the structure of each amino acid. Now, the way that this is going to work is that it has to do with ionizable protons. And interestingly, the backbone has ionizable protons in it. The amino acid backbone is what I'm referring to. So the amino group and carboxylic acid group both have ionizable protons. And these protons have respective pKa's. Now, textbooks in general are going to have a nice pKa table where you can refer to the pKa, particularly of the amino group of alanine or the carboxylic acid group of alanine. But for the purposes of this video, we're just going to look at the average amino group pKa and the average carboxylic acid group pKa for the 20 amino acids. And these values are about 9. So I'll say pKa is about 9 for the amino groups. And the pKa of the carboxylic acid group is about 2. And the way that we're going to assess how pH is impacting whether or not these ionizable protons that I've highlighted are going to be present or absent is by this rule of thumb. If the pH that we're using, here it's 7, is less than the pKa of the ionizable proton in question, then the proton is going to stay. On the other hand, if the pH is greater than the pKa of the ionizable proton in question, then the proton is going to leave. So here we have a pKa of 9 that we are comparing to a pH of 7. So 7 is less than 9, which means that we're in this first scenario, and the proton on the amino group is going to stay. When we look at the pH of 7 in comparison to the pKa of 2, 7 is greater than 2, which means that we're in this second scenario, and the proton will leave. So the way that that's going to impact the structure is that this hydrogen actually isn't there. And instead, we're going to have this oxygen that has a negative charge to it. Because we've used these as the average values for the amino group and the carboxylic acid group, and we know that this backbone is shared for each of these amino acids, we can modify each of the structures to represent the negative charge at the carboxylic acid end for all of our amino acid backbones. And so that's what I've done here for the sake of time. Now, what we see here in blue is the pKa for each ionizable proton that is a member of a side chain. So using our rule of thumb again, we know that our pH is 7, and the pKa here is 8.3. 7 is less than 8.3, which puts us in this first scenario, where the proton will stay. In the case of aspartic acid, looking at 7 as the pH and 3.9 as the pKa, 7 is greater than 3.9, which puts us in the second scenario. The proton will leave. So I'm going to zoom in just so that I don't mistakenly erase something I'm not intending to. And I'm going to give the carboxylic acid a negative charge on that oxygen. So you may have heard this amino acid referred to as aspartate instead of aspartic acid, and that's because we have the carboxylate group there. We can do the same assessment for glutamic acid, where we're looking at a pH of 7 compared to a pKa of 4.3. 7 is greater than 4.3, which means that, like in the case of aspartic acid, the proton leaves. So I'm going to erase that hydrogen and give it a negative charge to give us a carboxylate in our side chain. So you may have heard this as glutamic acid, or you may have heard this as glutamate, because at physiological pH, we're going to have deprotonation because the pH is greater than the pKa of that ionizable proton. There are some amino acids that don't have ionizable protons in their side chains. And so we're going to move past those, like phenylalanine and glycine, and jump over to histidine, which does have an ionizable proton. The pH of 7, we are comparing to the pKa of 6, where 7 is greater than 6, which means that histidine is generally not going to have this hydrogen when we're looking at pH 7. So I'll erase this positive charge, and the side chain then is neutral. Lysine is our next amino acid with an ionizable proton. And when we compare the pH of 7 to the pKa of 10.5, 7 is less than 10.5, which puts us in this first scenario, and the proton will stay. So at pH 7, we're going to maintain our positive charge in the side chain of lysine. Moving on to our next ionizable proton, we see that is in arginine side chain, where 7 is our pH that we're comparing to 12.5. 7 is less than 12.5, and if the pH is less than the pKa, that means the hydrogen stays. So we're going to keep this positive charge on the nitrogen and have a charge attributed to the side chain. The pKa of serine is 13 of the alcohol proton. So 7 compared to 13, 7 is less than 13, which means that the proton will stay. And so we'll keep the alcohol there. Threonine has the same pKa, so 7 compared to 13 again is the pH less than the pKa, so the proton will stay. Lastly, we have tyrosine, comparing the pH of 7 to the pKa of 10.1. 7 is less than 10.1, and so we will keep this proton as well. And so I've chosen 7 for this example, but you can certainly choose other pHs and get an idea of how that is going to impact whether or not a proton is going to be present or absent by comparing the pH and pKa values.